Differentiation in Eudorina elegans. So, as our background, we see our multicellular organisms usually have division of labor. For example, the bullbox cardii, they have somatic cells which help with movement, and the gonadea which help with reproduction. And these specialized cells, which again is specialized cells equal division of labor, each cell can focus on their own task. More than one thing can be done at one time. In natural selection, which is a process which causes traits to become more common, usually due to improved survival and reproduction, they're the dominant one in that area. For example, there was the dark mice we talked about. They survive better on the dark rock than light mice on dark rock because they blended in better. So linking that to multicellularity, they were favored due to the size. Small predators could not eat the bigger prey. And the Bulbakin, as I read, the clustering together, it gave them advantages. They were able to be bigger, so the ones on the inside are protected from the predators compared to the ones on the outside. My predictions. So research question was, how does the response to stress change when algae evolved a second cell type? My hypothesis for that was that when the Eudorina experienced a repeated cold shock, they will still increase in the number of differentiated cells when getting a second cold shock. My more specific prediction is when the experimental subgroup 2 is a secondary cold shock, the differentiation in cells will be higher than the differentiation in cells of the experimental subgroup 1, which received no secondary cold shock. So we used Eudorina elegans. So they are multicellular algae, and they are undifferentiated usually. They do have this special thing that allows them to temporarily be differentiated, but it's not genetically inherited. It's not passed down through the cells. What makes them this, given the specialization, is the presence of stress. So maybe things such as predators, in our experiment was cold shock, limited food resources, things like that. For this, they actually represent an early flexible form of multicellular division of labor. So our procedure. We first started, we had an experimental group and a control group, two different groups. The experimental group received a cold shock in an ice chest for 2 hours a day for 5 weeks. Our control group remained at room temperature. We set it next to the ice chest for 5 weeks for 2 hours as well. At the end of each 2-hour treatment, we would transfer 2 milliliters of the algae into a new tube with 8 milliliters of the standard Bullbox medium, S&B. And in between each treatment that we did, it was maintained in a walk-in chamber at 25 degrees Celsius, and it was illuminated under a 24-hour grow light. After that 5 weeks was up, we split our treatments, each treatment, into 2 smaller groups, creating 4 groups total. But we did the same thing as we did before. We had 2 milliliters of each treatment into new tubes with 8 milliliters of S&B. Our 4 groups were E1, experimental 1, E2, C1, control 1, and C2. The groups were then returned to the walk-in chamber under the same conditions they did before. 2 days before we collected data, the 2 subgroups received a secondary cold shock. Same conditions, 2 hours in the ice chest, and the experimental one, and then the control group was also this time put in for the ice chest. We used E2 and C2. And then once that was done, 2 days later, we collected our data. We collected it through photos. We had 160 total. There was 40 for each subgroup. So we had specific requirements. So our size should be changed every 8 minutes. Occasionally we changed them sooner when we needed to. When we stopped finding them, we would switch out. Our photos had to be clear, and we had to have no biases. So the first one we came across that met all our standards, we had to take a picture of. So our standards today would be 80px or larger. And they had to have visible EMC. They had to be not too squished or too round. And they had to not be actively reproducing. So our results. For our control group, subgroup 1, we had 12 differentiated cells and 28 non-differentiated cells. For our control subgroup 2, we had 18 differentiated cells and 22 non-differentiated cells. For our experimental subgroup 1, we had 6 differentiated cells and 34 differentiated cells. For our experimental subgroup 2, we had 20 differentiated cells and 20 non-differentiated cells, a perfect half. Our control group statistics, our percent differentiated for control group subgroup 1 was 30%, and our control subgroup 2 was 45%. Our standard error for each group, control group 1 was 0.0725, and control group 2 was 0.0787. Our control p-value was 0.0384, and we used an alpha of 0.5. In our experimental group statistics, the percent differentiated for experimental group 1 was 15%, experimental group 2 was 50%. And our standard error for group 1 was 0.0565, for group 2 0.0791. And our alpha for that was such a small number, I'm not even going to attempt to say that, but it went very small. Our graphs here, for our control graph, we can see that there's not too high of a difference between the two, and our 95% bars overlap. For our experimental graph, we can see a much bigger difference in them, and our 95% confidence interval bars do not overlap over there. So for conclusions, we found that the secondary Kolchak procedure does, in fact, increase the differentiated cells. It ended up supporting our research hypothesis, so we could reject the null hypothesis. Our p-values were less than alpha for both groups, which meant it was statistically significant. Our differentiated cells can arise due to stress factors in natural selection. Proving it in our experiment, future directions. So if I were to repeat this experiment, I would probably choose to increase the stress factor past 2 hours a day, once a week. I would change it instead to 2 hours a day, once a day. So for every day for 5 weeks. I had some new questions that arose during this experiment. The first thing was there any allergies that had more of a permanent differentiation? Ones that didn't, that were in our secondary cold shock group that were there before we cold shocked them? Were there some cells in the control group that were differentiated? Why were there some cells in the control group that were differentiated even though the stress factor was not introduced? So we had, I go back to our groups. Here, our control groups had a lot more differentiated cells than what I was expecting. It'd be nice to know exactly what caused that and why. And why were there less differentiated cells in the experimental group that received no cold shock than the control groups? If we go back, we see our experimental subgroup that received cold shocks only had 6, which was our smallest number compared to our control subgroups that had a lot more. They were at least double and a little bit more. So I want to know exactly why that happened. What happened in this group that caused this difference? These new questions mean we get to do new experiments. I'm not sure what those experiments are just yet, but future projects, future experiments. For acknowledgements, I'd like to acknowledge the University of Arizona. They were able to provide the materials for this experiment, our tubes, the space. I'd like to thank the mitochondrial lab at the university. They provided the Eudorina for the experiment. Without them, we couldn't have even done this experiment. And not to be super narcissistic, but I'd like to acknowledge myself for doing all of this myself as well. Any questions?